The aim of this experiment is to study binary fission in amoeba, budding in yeast, and budding in hydra with the help of prepared slides. So material required is a compound microscope and already prepared slides of binary fission in amoeba, budding in yeast, and budding in hydra. The word binary means two. So binary fission means when one organism divides into two organisms or two daughter individuals. So you can see in this image that this is the parent hydra. So when the process of binary fission begins, the nucleus first elongates and then divides into two, followed by the division of the cytoplasm, resulting in the formation of two daughter amoebae. Let us now look what would this look like when seen under a compound microscope. So you will see this happening in the slide that has been prepared. It might show a slight variation. Maybe in some slides you might see the nucleus dividing, in some slides the stage is much more advanced, as in this case. Here you can see the nucleus has already divided and now the cytoplasm is dividing into two. Next is budding in yeast. Yeast is another organism which reproduces by the process of budding. Budding means development of a small little outgrowth on the parent organism, which later develops into a new individual which is capable of independent existence. But in case of yeast, the new daughter individual that is formed stays attached to the parent cell, resulting in the formation of a chain of yeast cells, which are also known as the chain of buds. So let us see how this looks under a microscope. Under a microscope, you will see this. So the light structure that you can see in the center, this is the nucleus of one yeast cell. If you look at this one, you can see a chain of cells here. I can identify 4 cells which are joined end to end. So this has been formed as a result of budding in yeast. Next, we'll be talking about budding in hydra. This slide shows how budding happens in hydra. Hydra is a multicellular organism which reproduces by the process of budding. So in the first image, you can see a small outgrowth here, which then develops into a baby hydra or the daughter hydra, which grows and finally detaches itself from the parent organism. Let us see how this would look under the microscope. So if seen under a microscope, you will see structures like this. So this is the parent hydra with its tentacles, and this is the bud that has been formed.